Hi everyone. In this video, we will be talking about cellular respiration, a way to basically make energy in cells. So essentially what cellular respiration is that basically cells need glucose, it obtains it and makes ATP out of it. So in most cells, the way to make ATP or energy is through a process called cellular respiration. It's where glucose is broken down and basically the energy is harvested from the electrons used to make ATP from ADP and the inorganic phosphate group. So plants do this through uh, photosynthesis. We'll talk about that in the next video. And then with the many other organisms obtain glucose from food, and the glucose is further stored as glycogen or starch. And generally, they have enough ATP to sustain them from like 30 seconds to a few minutes of activity. Since ATP, like we talked about, is very unstable, the cells are making it constantly. So what actually happens when a glucose molecule's bonds are broken apart? When a glucose is broken down, basically the carbons within that glucose molecule ends up oxidized. So what happens is you end up adding oxygen to it. And what happens is they form carbon dioxide with water, basically. And it's basically that the oxygen is the final electron acceptor in the process when it becomes reduced to form the water that's there. So glucose oxidation itself is a series of carefully controlled redox reactions, which basically harvest the high energy electrons and hydrogen ions or protons in this case. And the energy itself is used to synthesize ATP from ADP and the inorganic phosphate. Basically, all of these reactions together make up what is called cellular respiration. So one of the other questions we kind of think of is how do you actually capture the amount of high energy electrons that you can end up using them later on in like the processes that needs to happen. And the first thing is the idea of electron carriers. These are very important molecules that are involved in cellular respiration and photosynthesis. They basically shuttle the electrons to the transport chains where the ATP is produced. It also involves like molecules like nicotinamide, adenine dinucleotide or NAD, where it ends up in an oxidized state like NAD plus, or a reduced state where it gets a hydrogen added on, which is like NADH. Similar scenario with FAD and FADH2. So the electron carriers themselves, what happens is that the reduced NADH carries 2 electrons and 1 hydrogen ion, more than the NAD plus. The reduced FADH2 carries 2 electrons and 2 hydrogens more than FAD. So that's just some things to keep in mind when we talk about electron carrier transfer proteins, transport electrons, sorry, to basically harness that energy uh, in to bring them towards areas where that energy is harnessed. So now electron carriers can also carry hydrogen ions. It's basically when these two are reduced to NADH and FADH2. And what happens is that they also have picked up hydrogen ions from the surrounding solution. So what happens is that the hydrogens in these names tell you they've been carrying electrons and hydrogens, both of which will eventually be used in the process of ATP synthesis through the electron transport and oxidative phosphorylation processes. So the structure of the mitochondria itself, so now that we've kind of talked about that, we're going to talk about where this actually occurs. So the mitochondria itself is basically, to most people, is known as the powerhouse of the cell. If you want a more in-depth detail about the mitochondria, you can check out this video. But essentially, these are some core parts of the mitochondria itself, where the cristae is the sac of membranes here, joined to the rest of the inner membranes that you see here. Then there's the matrix, the cristae, the inner membrane, the intermembrane space, and the outer membrane. So the intermembrane space here, and the outer membrane. This is another view of the mitochondria where the mitochondria itself has its own DNA. It has its own little ribosomes and all of the different spaces that we talked about earlier. So in cellular respiration, what happens is it is basically a set of reactions that basically produces ATP in the electron transport chain. It has 4 different steps, including glycolysis, pyruvate processing, citric acid cycle, the electron transport, and the chemiosmosis. And we'll talk about these more in depth in the next few slides. So glycolysis itself is the processing of glucose into pyruvate. Pyruvate is one of the first set of molecules that are involved in cellular respiration. So this is the first step. It's basically all of the enzymes that are required are present in the cytosol itself. In this scenario, oxygen itself is not required. In glycolysis itself, it's broken down into 2 pyruvate molecules where ATP is produced through a process called substrate level phosphorylation. That's what is happening here. And the NAD plus is reduced down to NADH, and it forms 2 ATP molecules here and 2 pyruvate molecules. Now, we take the pyruvate that was formed in that first step of glycolysis and goes into a step called pyruvate processing, which basically is the second step of this whole process. And if oxygen is present in general, the 2 pyruvate molecules end up entering this mitochondrial matrix that we were talking about earlier, and undergoes a series of reactions that end up leading to the production of what is called 2-acetyl coenzyme A, or acetyl-CoA molecules, basically. And during these reactions, another set of NADH is produced. And one of the carbon atoms from the pyruvate ends up oxidized into a carbon dioxide, basically. So this is what's happening here. It gets oxidized and we have a carbon dioxide as well. The NAD plus picks up the electron and the hydrogen atom ends up becoming reduced to NADH. That's how that process is occurring. Now we have what is called the citric acid cycle. This is the third step in cellular respiration. And what happens is that when the acetyl-CoA is produced, it goes into that cycle and it's located specifically in the mitochondrial matrix. Each of these acetyl-CoA molecules end up being oxidized into two molecules of carbon dioxide. And what happens is that the potential energy released from these acetyl-CoA breakdowns is basically used to reduce NAD plus to NADH, reduce the flavin adenine dinucleotide or FAD to FADH2, and then phosphorylate that GDP in there to become GTP. So this is like the sister version of ATP. This is guanine triphosphate instead of adenosine. This is guanosine triphosphate instead of adenosine triphosphate. So now there is a part to it that is referred to as the oxidative phosphorylation. Most of this occurs during cellular respiration itself. It's basically occurring in the last pathway itself, where this is the only pathway where oxygen is the input component. So it's like a substrate in all of this processes, and the only part of the part where it is being an input. And basically this pathway itself consists of the electron transport chain and chemiosmosis, which basically allows the generation of ATP. This hydrogen concentration gradient here does created by this electron transport chain ends up providing the energy and power to get chemiosmosis up and running. So now we are going into the fourth step of cellular respiration, where it's called the electron transport chain, where these high potential energy of the electrons carried by our two electron transport carriers, it's basically gradually decreasing as they move through a set of redox reactions. And what we see here is that these proteins that are involved, they end up making up an electron transport chain. And basically the oxygen is the final electron acceptor. And the transfer of electrons go from like protons and to the oxygen itself and form the water that's produced in this process. And ATP is finally formed again with from ADP and inorganic phosphates. So basically it's a whole set of redox reactions, which are basically oxidation and reduction reactions, where it occurs in an electron transport chain, where the proteins itself are increasingly electronegative. So the electrons end up moving down the electron transport chain to the oxygen, which ends up being that final electron acceptor, since it is the most electronegative of all of these components we see here. And when it goes down that gradient, down the ETC, what happens is that they end up powering that transfer of electrons or hydrogen ions across the membrane like this. And what happens is they basically set up an electrochemical gradient going in and out through the different complex. So that kind of leads into the idea of chemiosmosis. Basically, the job of the electron transport chain is basically to use that power of the electrons to pump proteins across the inner membrane of the mitochondria itself, from the matrix to the inner membrane space, and set up an electrochemical gradient using the hydrogen ions across that membrane itself. So this is all happening with like the inner membrane or the matrix of the intermembrane space itself. And once this proton gradient is established, the flow of proteins basically back down the gradient through ATP synthase. And what happens is it ends up allowing the ATP to form by giving it more energy to do that process. Now there is ATP synthase. It is also a motor that is basically like a molecular motor. So if you see anything that ends in with like ASE, that's an enzyme. And this enzyme basically acts like a molecular motor. And with the protons, what happens is that they flow through this F1 unit here and basically spin the rotor here, and then goes in and it goes through. It spins the F1 unit, and then the F1 unit spins, and it starts changing shapes and catalyzes the phosphorylation process of ADP, and the inorganic phosphate to ATP itself. So now with this, the chemiosmotic gradient, what happens is that the ions end up moving down their chemical and electrical gradient across that selective membrane. Remember, we were talking about our membrane that's very particular. Now, first, the protons are basically pumped against their concentration gradient across the membrane in the electron transport chain. And then the protons flow down their chemiosmotic gradient. So this is what a chemiosmotic gradient is. And what happens is that they go down their gradient electrically and chemically, and it goes down. That selectively permeable membrane. And then at the end, it basically gives you all the ATP that you would need to get things up and running. And compared to the rest of the cycles, most of the ATP or energy yield comes out of the oxidative phosphorylation process, which basically has about 25 ATPs. And what happens is throughout the entire process, you get 29 per glucose molecule that's basically broken down in this process. That brings us to the end of cellular respiration. In the next video, we will be talking more about photosynthesis and how plants do this whole process.